for those who are going to be, seeing our presentations from the virtual conference, I do have another talk on there specifically about participating in clinical trials, kind of what that's like in the rare disease space, some of the language to be familiar with, and kind of what that process looks like overall. Tonight, I'm going to be discussing a little bit about the impact of basic research in Duchenne, how to think about trials briefly, just in terms of some expectation setting, which I think is really important to do, if this is all very new, and then kind of going through what the drug development pipeline in Duchenne looks like today, what the therapeutic areas are, and kind of what potential treatments are available, and then just kind of ending on some of the resources that PPMD has for helping you to find trials. So a little bit of background on how PPMD has impacted research and drug development over the years. So to date, we've invested over $50 million into research and therapy development. So that's both basic science, that is preclinical science, so developing drugs with and investigating them in animal models, as well as tools to help accelerate drug development. And that's been given out over 260 different research grants. So projects aim to explore and understand Duchenne. Ultimately, by supporting that kind of research, that has resulted in us now having a very active pipeline. We have around 35 clinical trials active in Duchenne at any given time, which is really extraordinary for a rare disease. If you kind of look at the drug development pipeline in some of the other rare diseases, you just do not see this kind of breadth of potential therapies being developed. And to date, that has then resulted in our approval of our 3 U.S. drugs, 2 of which are mutation-specific, and then one of which is available for everyone, and I'll touch on those briefly as we go through. So all of that investment that we've done has really resulted in a wide array of therapeutic strategies in Duchenne. So different ways for us to attack this disease and develop new drugs that might be able to help treat all of the patients. And so I'm going to go through these different areas, but I wanted to talk really quickly about kind of what has led us to getting to these different therapeutic strategies, because I think it's important to understand when we are funding basic research or some of that preclinical research, why we do that. Especially if you're new to the area, I think sometimes when you see these announcements about some of the science being funded, it's not always easy to make the connection as to why. And by funding all of this basic research, what that has done for us is allowed us to get this kind of broad network of what's happening in Duchenne. So I don't expect you to read all of this. You don't have to know everything in here. But what I wanted to kind of focus on was over these years of investing in understanding Duchenne, that's let us go from a basic understanding of a mutation in the DMD gene that is leading to no functional dystrophin, to understanding what all of these downstream issues are that come along with the disease. And so by understanding all of these different areas, that actually opens the door to discovering new druggable targets. So even if we can't address necessarily the lack of dystrophin, we can address things like the chronic inflammation or dysregulation of calcium. And it also opens the door for researchers that maybe aren't in the Duchenne space or companies that aren't in the Duchenne space. If they have products that fit somewhere within these pathways, by investing in this kind of research and publishing on it, it kind of signals to those people that maybe they have a product that can be brought into our drug development landscape, which I think is really important, and again, why we have such a robust pipeline currently in Duchenne. So really quickly, I'm going to talk about why we do clinical trials. The purpose of a clinical trial is for us to understand if a drug that is being developed is safe and efficacious. So Ryan already kind of talked on to some of these points earlier with kind of the generation of the FDA as a regulatory body, and it's so that way that we know that the products that we end up delivering to these children are providing them with benefits and aren't going to have a bunch of negative side effects. Now, something to keep in mind as you go through this process, I always like to remember that the reason why we are testing these products is because we don't yet know if they work. If we knew that they were going to work, we wouldn't have to do clinical trials. So clinical trials are still a type of experiment. These drugs don't get to humans first without going through a lot of testing at the bench, going through a lot of preclinical and animal model testing. But that preclinical testing is not, one-to-one. A mouse or a dog is not a one-to-one comparator to a human. And so we do have to go through these steps of testing the drugs in humans to make sure that they're safe and that they're working. And also, this is kind of the other challenge with drug development, is that there is a lot of failure in drug development. And so this is kind of just something to keep in mind. We like to talk about hope and not hype, and that this is a challenge. Now, we've had fairly good success within the Duchenne landscape. Like I said, we have 3 approved drugs in the US. There's some in the EU and Japan. There's some other products that have been approved or conditionally approved that are being tested in the US. So there's a lot of hope. But we do have to go through all these different phases of clinical trials to get to an approval. So this does take time. There is risk for failure. But where we're at currently in Duchenne is really exciting. We have a lot of trials that have made it to these phase 2 and phase 3 stages, and that's really promising and is a good indication that we're on the right path. So to come back to the therapeutic strategies, what I'm going to do now is kind of go through these different areas, and we're going to talk a little bit about what is currently being done in Duchenne with regards to each of strategy. Now, for a lot of these, there are going to be presentations available from the sponsors. Sometimes I'll say sponsors, sometimes I'll say companies, by the way. That's just interchanging the same word to say the pharmaceutical company that's developing these products. They have presentations that you'll be able to watch, and then, of course, we'll have them available during our live sessions to ask questions. But we're going to go through these kind of different areas and how we're thinking about treating Duchenne by targeting these areas. So the first one is dystrophin restoration and replacement. And so, as Anne just talked about, the cause of Duchenne being a mutation in that the DMD gene causes an absence of the dystrophin protein. And so one of the strategies that we can take is, how do we either restore the production of that dystrophin protein or replace that dystrophin protein? And there are a number of strategies currently in development. One of the ones that you're going to hear about, and there's a lot of excitement with, is the gene therapy strategies. So just a very quick kind of primer on what the components of gene therapy are. The idea with gene therapy is we take a piece of DNA that could be beneficial, we package it with something called a promoter, and that's a piece of DNA that helps control where that gene is going to be turned on. So in this case, given that Duchenne is, the lack of dystrophins affecting the muscle, we want to make sure that we are taking that DNA that's going to be helpful and that we're turning it on in the muscle. And we package all of that into a vector, which in this case is a virus called adeno-associated virus, or AAV, that you'll hear a lot about. And this can be delivered throughout the entire body, gets into the muscle tissue, as well as some other organ tissue as well, and allows for that whatever gene we're interested in to be delivered to that tissue. In this case, like I said, it's muscle. And so our current gene therapy strategy is around replacing the missing dystrophin protein. So as Anne said, the DMD gene is one of the largest in the human bodies. This is a schematic of the protein. So these are different domains of the protein. And with the strategy for gene therapy currently is to make a miniaturized version of this full-length dystrophin protein and get that into all of the muscle tissue. And the reason why we have to go for these mini and micro dystrophin constructs is the AAV is a tiny virus, and it doesn't have a lot of storage capacity, essentially. You can only pack a little bit of DNA into there and deliver it. So through research that has been done, so to get here, what happened was we looked at patients who have Becker muscular dystrophy, as Anne noted, tends to be a less severe dystrophinopathy. And in Becker muscular dystrophy, those patients tend to have a truncated or a shortened version of the dystrophin protein, and they might still be ambulatory well into their 6th and 7th generation. And so scientists looked at the proteins that those Becker patients had and then tried to mimic them in these mini and micro dystrophins. And so that's what we're delivering to the cells in the current gene therapy strategies, in hopes that we can help restore some of the function of the missing dystrophin. Now, of course, it's not going to turn a Duchenne patient into a Becker patient, but it can really help ameliorate a lot of the effects that we see of the disease and help slow its progression. Other strategies for restoring dystrophin production falls into some of the exon skipping that you'll hear about. And so Anne touched on it briefly, and I'm going to have a little schematic after this. There's a lot of different terms that are talked about within exon skipping. You'll hear the number of different acronyms. You'll hear ASO, PMO, PPMO, U7S RNA. So these are all different technologies aimed at doing the same thing, which is to take an out-of-frame mutation, put it back into frame, and create a truncated form of the dystrophin protein. We also have the nonsense mutation read-throughs, and so for those individuals with Duchenne who have a premature stop codon, there are strategies to basically ignore that period that's showing up in the sentence where it's not supposed to be, to ignore that and continue to read the sentence, to use some of the language that Anne just showed. And there's also some cell therapy strategies looking to restore or replace dystrophin. So a little more on the exon skipping, and I think did a nice job of covering it, but just to help visualize it. In a normal kind of healthy patient, we look at having all the exons. When they go to the mature mRNA, they all line up like puzzle pieces, and you get a normal full-length dystrophin. And someone who has a deletion, so in this case a patient who has this exon 49 and 50 deleted, what happens when they go to the mature mRNA, the pieces of the puzzle no longer fit together and we don't get any production of dystrophin. The exon skipping therapies essentially say, we're going to try to skip one of those exons that's causing the pieces to not line up. And by doing that, we can restore the reading frame and we get this nice shortened functional copy of a dystrophin protein. And so there are a number of companies that are developing strategies for that, as Anne alluded to. And these are all mutation specific. So we have 2 approved therapies for this. So Exondys 51 and Vyondys 53 are 2 approved therapies for those amenable to exon skipping for exons 53 or 51. And then in development, so still in clinical trial phases, but in phase 3s, phase 2s trials, we have skipping for exons 45, 53, 51, and then of course we have for the nonsense mutations the product from Adalorin, and you'll be able to learn about all of these over the coming weeks. For reducing inflammation, so one of the prominent features of Duchenne is there is this chronic inflammation, because with the breakdown of or the instability of the muscle cell membrane, we have kind of all of these other things going on. There's influx of calcium, and just there is this state where the muscle is inflamed, and what our current standard of care is to treat that with steroids. So you also see glucocorticoids. So these are strategies that look to minimize that inflammation. Some of them are being developed to replace steroids. One of the complaints that we often hear from families is that although steroids have a lot of benefits in terms of delaying disease progression milestones, it's longer periods of ambulation, longer or better respiratory function, there are side effects with steroids. That's the weight gain and cushingoid. Sometimes can be, different temperament in terms of mood. And so some of these strategies look to replace the use of steroids, and some of them look to work in concert with steroids. And so we have a whole host of products that are being developed to address inflammation in Duchenne. One thing I do want to note here, so I put over here whether or not steroids are, a requirement for the study. In most clinical trials in Duchenne, steroids are a part of the inclusion criteria, meaning you need to be on a steady dose of steroids as part of standard of care in order to participate in that trial. In these clinical trials, where they're also looking to reduce inflammation, there's often, they want patients who are steroid naive, so patients who have never used steroids, because they're going after the same target. So if the steroid is trying to reduce inflammation and the product you're developing is trying to reduce inflammation, it's hard to detect which of those two drugs is responsible for any benefit we see. And so in these instances, they may say that we need patients who haven't taken any steroids. And so if you are newly diagnosed, if this is still very early on in your treatment, and maybe your child isn't on steroids, these are some trials you may also consider looking into, since they have that requirement to be steroid naive. We also have some trials listed here that aren't taking place in the United States. The tamoxifen is an EU-only study. This ATL 1102 was developed in Australia, and they are beginning a trial in the EU as well. So next I want to talk about some of the areas that we deem as muscle growth and protection, and then kind of combating fibrosis. And so one of the issues within Duchenne is, as there's this chronic inflammation, it does impact the regenerative capacity of the muscle. So as Anne showed in that kind of very first image of looking at kind of a healthy piece of muscle tissue, and then one in Duchenne, we saw that over time that there was that replacement with bifatty deposits and scar tissue that we refer to as fibrosis. And so these are strategies that look to either inhibit the development of that fibrosis, or in addition to inhibiting fibrosis, they may have other effects like some anti-inflammatory effects, or helping promote more muscle regeneration. And so these are ways to also protect and grow the muscle. And so we have a number of compounds, again, that fall within this area of either anti-fibrotics or things that are helpful to restore muscle growth and protect the muscle. And again, like I said, we have these at various phases of development. In this cohort, we see that a lot of these trials do require steroids. I do want to point out, I'm not sure if we have any newly diagnosed families with a Becker diagnosis. This product, Javinostat from Italfarmaco, is one of the few drugs that's also currently being looked at and investigated in a Becker population, which is interesting. A lot of these compounds that are downstream may also be work for patients with Becker, although typically they are first tested in Duchenne, which is the more severe disease. We also have drugs that are targeted at regulating calcium and improving the mitochondria. So within Duchenne, because we have that leaky muscle cell membrane, calcium is no longer regulated properly. In a cell that has its normal cell membrane, there's channels for calcium, but this is dysregulated in Duchenne because of all of this damage, and so that causes a host of other issues, including also dysfunctional mitochondria. And so by addressing things like the calcium balance, we may be able to improve the function of the cell. We may also have an impact on the mitochondria, or we can also look at strategies that just try to improve the mitochondria. So the mitochondria help produce energy for the cell and help it carry out its normal functions. So by either taking control of the calcium or bolstering the function of the mitochondria, we might be able to improve the quality of the muscle tissue to help prevent some of the necrosis and the fibrosis that we see in the muscle over time. And so we have a number of strategies, again, that look at addressing these. The top three here are all strategies that look specifically at improving mitochondria. And so you'll so through a process called mitochondrial biogenesis. So trying to generate better and more mitochondria, as well as we have a product, ramipride here, which is looking at trying to help regulate that calcium balance in the cell. And again, just to show that for all of these different areas in Duchenne that we've identified as issues, we have a number of different investigational products that are in development trying to address these aspects of the disease. And finally, we have the heart, and as we always like to say, the heart is a muscle too. These same pathways that we see, the fibrosis, inflammation that contribute to the weakening and the damage of the skeletal muscle, do play a role within the heart muscle as well. But we do have a challenge that within cardiac development of cardiac drugs that we don't see within the skeletal muscle, and that comes down to something called the endpoints. So in some of the drugs that look to improve, function of the skeletal muscle, they can take within, the boys, and they can test something like their ability to improve on something like the 6-minute walk test. So, how far do they walk in 6 minutes? And we can assess whether a drug is working by saying, they've improved on their time, or maybe they've stabilized and they haven't had any decline in a long period of time. And we can use these kind of markers to say that a drug is having a positive effect. With the heart, it's a little more challenging. The endpoints that we have currently aren't as predictable. Some of the things that we use, like ejection fraction used to monitor, that doesn't follow the same kind of trajectory in natural history that some of the skeletal muscle follows. So it can be a little harder to test. We do have one product that is currently in development looking at addressing the heart, and specifically by minimizing inflammation and fibrosis, trying to strengthen the heart. Now, there are other, medicines that are available for the heart. I'm sure if there was a discussion yesterday, there's beta blockers, ACE inhibitors, spironolactone, eplerenone. But in terms of new products in the pipeline, we have currently this ifetroban. And so I know that was a lot of kind of what is in the pipeline currently. We do have some tools to help you kind of sort through this to get a sense of where these products are in development, and whether there's trials open and available to you. So we have our interactive drug pipeline, which characterizes and tracks all of the different investigational products in Duchenne, what phase they're at in development, along with some language that we hope helps you to understand what the expectation is in terms of what that drug is going to do and the potential benefit it may provide. We also have our trial finder tool, which is a search tool that we've developed that helps kind of categorize those clinical trials by a number of different factors. So you can search by age, you can search by therapeutic area, whether there's trials in the US, and use that to kind of direct your search for trials you may qualify for or your child may qualify for. Of course, we have the Duchenne registry, which is a great resource. When your information is in there, when we do targeted recruitment, you'll only be recruited for trials that you may qualify for. And then, of course, one of the most important aspects that we really think you should utilize is your care team. Your doctors are going to know a number of these trials that are ongoing. They're going to know if they're taking place at your site or if they're taking place at nearby sites, and they can really help you navigate this. And then clinicaltrials.gov is the kind of web resource that all of the sponsors are required to post information to. I would suggest that if you're using clinical trials, you use that with maybe your care team to go through with that. It can be a bit confusing. There's a lot of information there, a lot of good information, but if it's your first time kind of diving into the clinical trial landscape, that may be something where you want someone who has a little bit of expertise to help you kind of navigate that resource. And so I think at this point, I'm happy to take any questions. I don't know if it's